Hi, thank you very much for this invitation to present the work we're doing in the laboratory to try to understand the intersection between the microbiota and the immune system. So as many of you do know, of course, the microbiota that colonize all barrier surfaces has been shown to play a fundamental role in the development and function of the immune system, including the ability of the host to mount protective response to pathogen, vaccine, and recently really nice work demonstrating a role for the microbiota as an adjuvant in the context of immunotherapy. This regulation of these responses can, of course, also lead to inflammatory disorders, including IBD, autoimmune disorders, and allergy. However, despite this broad understanding of the fundamental role of the microbiota in the control of the immune system, we're still lacking in-depth mechanistic understanding of the mode of communication between this microbe and the immune system. In particular, a question that became a center of the attention in the laboratory is to try to understand what is unique about the way the immune system sense and respond to the microbiota. And the reason we decided to explore this question because we felt that at the time, most of what we understood about how the immune system functioned really came from the seminal work that was done by Genoway and Polly Matzinger and many others, highlighting the importance of inflammation and tissue damage in the induction of adaptive responses that can in turn go back to the tissue and control the pathogen. However, most of the encounter of the microbiota with the immune system really occur in complete absence of inflammation. It's a very homeostatic form of interaction. Nonetheless, there is clearly induction of both T cells and B cell responses that accumulate within the tissue. So over the last few years, we have begun to explore two questions. The first one is what could be unique about a class of immune responses that is not really here to control the microbe that live outside of the body surfaces. And the second, what is really the original thing, the mechanism by which this microbe are able to engage the immune system in such a quiescent way. I'm going to go very briefly on this first part that we have actually mostly published to actually discuss some recent work that we have where we have uncovered a novel element that appear to be fundamental for the control of host microbiota interaction. So although we do work within the GI tract, the lung, and other barrier surfaces, the work that we do to try to understand this specific question is in the skin. And the reason behind it is, of course, because of the beautiful work that was done in the lab by a very talented graduate student, Judy Naik, when she uncovered a fundamental role for the microbiota in the control of the immune system. And what is unique about this compartment is that despite the fact that this microbe are present at low biomass and in very specific structures, such as hair follicle and sebaceous gland, these microbes are able to potently control the immune system. So we believe that this approach of using a tissue that is accessible, but also with a low biomass of microbe may really represent an ideal strategy to understand fine mechanism of interaction. So over the last few years, really work that has done by numerous investigators in the laboratory, most of them have started their own group right now, highlighted then the class of immune response that is induced in response to microbiota is quite unique. These cells are generated in complete absence of inflammation. There is no neutrophils, no macrophage in terms of induction and accumulation of the cell. However, both CD4 and CD8 T cells have the ability to embed themselves in the epithelium, such as in this case, CD8 T cells in response to Staphylococcus epidermidis. These T cells, when they go in the epithelium, are able to communicate with keratinocytes, via IL-17 in particular, thereby enhancing the ability of the tissue to promote antimicrobial defenses. But what we also found that was quite unique about this class of immune responses is not only it has this bystander effect on antimicrobial defense in the host, but it's also able to promote tissue repair. And this was demonstrated in the context of skin injury model. So the cells are highly plastic and can adapt their function and really produce large amount of type 2 cytokine in the context of injury. More recently, work that is done in laboratory by Michel highlighted the fact that in addition of the tissue repair and keratinocyte function, those cells are also fundamental in controlling the ability of sensory neuron to regenerate in the context of injury. So it's a class of immunity that is really not here to control the microbe that is exposed to staphylococcus in this context, but to enhance antimicrobial defenses very broadly, and also to promote other physiological aspects of tissue function, namely tissue repair. So the question we had is really to try to understand the mechanism by which these responses are induced. And this led us to revisit some of the work that we obtained a few years ago in the context of work that was done by Samira Tamutunyo, and she was in the laboratory. When she found that if she applied a microbe at the surface of the skin, and these animals have their own microbiota, what she was able to show is that a very surprising type 1 response is antiviral defenses. So this is paradoxical responses where association with the microbe does not cause any inflammation, but has this actually really strong antiviral defenses program. So this was actually not new in retrospect, because work that was done first by David Ardis in January, but also subsequently by Akiko Iwasaki and many others, highlighted that the gut microbiota has this ability to broadly promote antiviral defenses at distal tissue. So there is this known association between the microbiota and the ability to promote antiviral defenses across the system. And this actually led us to revisit another fundamental aspect of host physiology, which is the fact that, of course, we have an exogenous microbiota that colonize all the body surfaces, but what we also have is an endogenous virome. Our genome is composed of 10% of endogenous retrovirus that have embedded themselves in our genome and being deactivated over time or neutralized or controlled. Nonetheless, some of these elements can maintain transcriptional activity and have been shown to biological function. So this actually led us to hypothesize and maybe a link between the exogenous microbiome and the endogenous virome that could explain this pattern signature. And this hypothesis was not new neither. In fact, the first person that actually thought about this question was George Castortes, who actually showed a few years ago that if you utilize an animal that is germ-free, there is a change in the basal expression of endogenous retroviruses in tissues. He was able to show also that if you stimulate B cells or macrophages with TLR ligands, you can have an upregulation of certain class of ERVs, so really downstream of macrobal stimulation. And finally, very fascinating, he also found that ERVs are highly tissue specific. So we decided to actually revisit this question in the context of this host microbiota interaction. And this was done in the laboratory by Jalma and Sid, who decided to assess keratinocyte response. That is, of course, the first line of response to microbiota. So animals were applied with staphylococcus at the surface of the skin. These are not germ-free mice, these are actually animals that have their own microbiota. And they purified the keratinocyte and the RNA-seq and were able to show that indeed there is an upregulation of ERVs within the tissues that can be actually shown in the right. So, of course, these ERVs can be subsequently evolving in the cells in a different kind of path and can be potentially reverse transcribed and lead to the production of double-stranded DNA that could potentially accumulate within the cytoplasm and therefore be sensed by the innate system as an alarm system. So to test this possibility, we utilize the drug that is utilized in the context of antiretroviral treatment that blocks the ability of the host to just have a reverse transcriptase activity. This, therefore, will prevent the ability of this element to be reverse transcribed by the DNA. So as you can see on the right, in the control mice, there is actually an upregulation of staphylococcus. When you put staphylococcus at the surface of the skin, there is an upregulation of T cells in the skin compartment. However, if you have treated the animal with an anti-RT, there's a profound downregulation of the ability of the mouse to mount a T-cell response against the same bacteria. This can be visualized in a more spectacular way using imaging, where you can actually see the epidermal layer that is visualized in blue post-staphylococcus association, and the massive accumulation of CD4 and CD T cells in the epidermis. These cells are actually antigen-specific to the microbe. If you treat the animal with anti-RT, you can see that there is a shutdown of the ability of the cells to just be generated and accumulate in the tissue. So blocking reverse transcriptase activity of the host, and thereby the ability of this element to become DNA, has actually a profound impact on the ability of the host to develop immune response to the microbiota. So we had shown before that this response to the microbiota could actually, the T-cell response to the microbiota can activate keratinocyte in a way that promoted not only antimicrobial defenses, but also tissue repair. So we revisited this question in the context of this antiretroviral treatment. And as you can see on the first panel here, an unassociated mice, associated mice with staphylococcus developed clusters of keratinocytes that are characterized by a high level of antigen presentation potential as MHC class 2, but also have actually interesting molecules that are linked to wound healing as well as antimicrobial defenses. If the animal has been treated with an antiretroviral, this ability of the keratinocyte and the specific cluster is really no longer developing in the tissues. So clearly, not only anti-reverse transcriptase has actually an effect on the ability to mount T-cell response, but also on the downstream consequence of this response. So to assess the possibility that this was indeed functionally linked to the ability of the animal to develop tissue repair, what we did is actually to do a model of wound that is actually characterized by a punch biopsy in the back and the measurement, a very fine measurement of the repair process via the assessment of the length of keratinocytes, the epidermal tongue, that proliferate and migrate within the bed of the wound. If you apply staph epi, as we have shown before, there is an acceleration of this process, lengthening of the epidermal tongue. However, if you have treated the animal with an antiretroviral, this ability of the bacteria and T-cell response to this bacteria to induce tissue repair is actually inverted. So blocking reverse transcription in the tissue limits the ability of this microbiota to promote this positive effect on host physiology. So of course, DNA that will be aberrantly accumulating in the cytoplasm will be sensed by the canonical machinery cGAS sting. To address the role of cGAS or sting in these processes, we apply staph epi at the surface of the skin, looked at T-cell responses, as we have shown before, and repeated the same experiment in an animal that were deficient in cGAS or deficient in sting. And as you can see here, once again, the ability of the animal to mount T-cell response to the microbiota is inhibited in absence of this pathway. But of course, trying to understand what will be the cell that are the determinant of these responses, we cross the sting flux mice in different cell subsets, and I'm going to show you here the data on keratinocyte that turn out to be the critical cells. So when you delete sting only on keratinocyte, you can see that at baseline, the level of CD8 T cells is already diminished, really showing that the tonic accumulation of CD8 T cells in the tissue really depend on the ability of keratinocyte to express sting. When you apply staph epi, there is an enhancement of CD8 responses, and you can see here that this is profoundly decreased if the animal does not express sting on keratinocyte. Similar response on mate cells and other population of cells we have shown to be induced by the microbiota and also linked to tissue repair. The baseline accumulation of the cells is decreased, and so is the ability of the cells to be increased post-staph epi association. And this response is even more dramatic in the context of gamma delta and other population of lymphocytes we have shown to be induced in response to microbiota. So clearly, keratinocyte expression of sting is a critical determinant of the ability of the host to mount broad response against the microbiota. So what we found at this stage, and what I would like to summarize before going to the next step, is that the microbiota appear to interact with the immune system via yet another viral partner, which is actually the endogenous retroviruses. In response to TLR stimulation, I'm going to come back later to this point, the microbiota is able to communicate with keratinocyte, upregulate the expression of certain ERV that appear to be reverse transcribed, and these elements become cDNA that can be sensed by the cGAS sting machinery. These actually create those hotspots in the tissue that are not inflammatory, but nonetheless activated enough to engage the immune system, both locally and systemically via the dendritic cell activation. These together lead to the broad accumulation or proliferation of cells in the tissue that then come back or accumulate in the site, thereby promoting broadly host physiology and tissue function. But the next question we wanted to address is not only what is happening in the context of steady state, but what does happen in the context of inflammatory processes. As I mentioned before, the microbiota is of course central to host physiology, but it also has been shown to contribute to numerous inflammatory disorders. So to address the potentiality that this crosstalk in the microbiota and ERV can somehow be linked to inflammation, we utilized an approach we have utilized before. That is to change the diet of the animal and assess the ability to respond to the microbiota. What we have found a few years ago is if you take a microbe and apply them at the surface of the skin of an animal that is fed a normal diet, this microbe does not cause any inflammation. However, you take the same microbe in an animal that has been fed a high-fat diet, and the mice, this is sufficient to not cause inflammation. So really, it's remarkable that just a change in diet can transform an homeostatic relationship to inflammatory. And this is very much in line with what is known in human studies, in which the skin of individuals that are having obese or having metabolic syndrome is characterized by numerous inflammatory processes, in particular defect immune healing. So we utilize a fluorine system, as I mentioned before. Animals are placed at the surface of the skin, and we looked at the physiology of the skin. As I mentioned before, this process in animal fed a regular diet is highly homeostatic, no inflammation. However, if you do the same thing in an animal that is fed a high-fat diet, the same bacteria now cause inflammation, as evidenced by thickening of the epidermal layer. This can be visualized via keratinocyte by measuring the thickness of the ear, but also assessment of the proliferative capacity of keratinocyte and the thickening of the epidermal layer that you can see here, and can be also quantified by looking at K67 expression within the keratinocyte layer. So to try to see if under the settings what you have is a heightened ability of this ERV to be expressed, what we did is once again to do a sequencing and our RNA-seq of keratinocyte under the setting. And you can see here, looking at the different part of those ERV, GAG, propol, and envelope, there is really a massive upregulation of these elements within an animal that is fed a high-fat diet and associated with the bacteria. So there is really the synergy effect when the response to microbiota in an animal fed a high-fat diet lead to aberrant, excessive ERV activation. In right, what you can see, this was actually curated via the expertise of George Cassiotes. We underlined elements that can be potentially reverse transcribed in the tissue. So to go back to this point about reverse transcription and to which extent reverse transcription activity can be important for these inflammatory processes, we treated this animal with a reverse transcriptase inhibitor. And you can see here on the slide that if you have an animal that is fed a high-fat diet and associated with the animal, there is inflammation. But this antiretroviral can have a significant impact on the ability of the host to develop inflammatory processes. So clearly, inflammation caused by the bacteria under this setting is at least in part dependent on this reverse transcriptase activity and thereby likely this ERV activity. Going back to the cGAS pathway, we repeated the same experiment we had done before in animals that were cGAS, STING, or STING deficient in keratinocytes in animals that were fed a high-fat diet. And as you can see here, the level of accumulation of T cells is significantly decreased in all the animal deficient, and so is the pathology that can actually be seen in the context of this high-fat diet microbiota encounter. You can see a significant decrease in the cGAS mice, STING, and the animal that is deficient in cGAS in keratinocytes. So in conclusion of this part, what I wanted to say is, although we have now seem to have uncovered an additional layer of control post-microbiota interaction, and there's really this extraordinary multi-kingdom dialogue that really controls immunity to the microbiota. It appeared that ERV expression is highly required for response to skin commensal, and that the cGAS-STING pathway in keratinocyte is really the gatekeeper of both homeostatic response to the microbiota and response to these exogenous microbes. So I used to refer as the microbiota as the endogenous adjuvant, and I think we need to rephrase that. The real endogenous adjuvant of the system appear really to be the ERVs. So the model we have at the moment is the following one. We found that the microbiota in response to TLRs can actually engage TLR signaling in keratinocyte, and we have evidence of TLR2. This leads to upregulation of ERV that can be sensed with the cGAS-STING pathway, leading to these hotspots, the hotspots that can then activate the system and lead to the accumulation of T cells response that promote homeostasis. In the context of high-fat diet, there is a shift in threshold activation of the tissue. Response to the same microbe now lead to aberrant expression of this ERV that can be now sensed in an aberrant fashion by the innate immune system, leading now to pathological responses. So what we really think at this stage is that ERV appear to be a gatekeeper of the threshold activation of the tissue, and they really are the ultimate controller of the ability to develop either homeostatic or inflammatory responses to the microbiota. And we are now, of course, in the process of assessing in-depth mechanism of this interaction and how this interplay can also contribute to pathological setting in the context of not only diet intervention, but also genetic predisposition or tumor. But before I actually finish, I just wanted to give you a hint of direction we are taking in the context of human settings. And this is done in the context of a collaboration Kevin Hall at the NIH and done in the laboratory by Verena Ling, that's a bioinformatician. Kevin has actually generated an extraordinary cohort at the NIH that is highly controlled, in which individuals come for 4 weeks at the NIH and have either a ketogenic and a vegan in a sequential way. So the same individual will be actually assessed through different kind of diet. And part of this work has been published recently looking at the metabolic impact of this diet. And what we are doing currently is really looking at the immune system. And what we have found, which is quite remarkable, is that this diet have actually a distinct effect on innate and adaptive responses and have actually striking differences in the ability to manipulate the microbiota. But related to this talk, what I wanted to highlight is that what we found that has been actually assessed before, but not as much as it should have been, I think, is that really when you look at individual, one of the most variable gene expression appear to be within the endogenous retrovirus repertoire. Each individual has its unique signatures of ERV expression in the cells that circulate. And if you actually place this individual on a diet intervention, you can see that these individuals have actually a dramatic shift in ERV expression. These appear to be, in fact, the most upregulated genes in response to dietary intervention. So we are now in the process of really looking more in depth at the cell level, but it appeared that in human too, ERV are exquisitely sensitive to dietary changes, something that could also help to explain the role of actually nutrition on the control on the immune system, among many other phenomena. So I have, of course, to thank all the extraordinary people in the laboratory that have helped on this project. But I have also, and in particular for this purpose of this talk, I have to thank two people that are Jalma and Sid, together also with Verena, that are doing ongoing work in human, and all the remarkable collaborators we had over the last few years, and in particular, George Cassiotes, that really has been a wonderful collaborator in this study, and was really the first one to really think about the crosstalk in the microbiota and endogenous retroviruses. And I thank you all for your attention.